To this morning session. My name is Femke de Vrij. I'm from Erasmus MC, and in this session we'll have a lot of different things ranging from omics to GWAS to sponsor talks, and we'll start with Daan van der Vliet, who did his PhD in the groups of Mario van der Stelt and also Inge Huitinga. And I'm very happy that since last year he moved to my lab at Erasmus MC to work in this ICNS project, so the perfect combination of the chemical tools he already mastered and the stem cell modeling we do in our lab. So Daan, very much looking forward to your talk.
Thanks a lot, Femke, for the kind introduction. Yeah, so today I want to share my main project from my PhD time in the groups of Mario van der Stelt and Inge Huitinga. And I'm very fortunate that Bart already presented before me, because I basically want to pick up where Bart ended on the microglial states in MS lesions. So you've seen this information already. So multiple sclerosis is a very severe neurodegenerative disease characterized by these inflammatory white matter lesions. As Bart already mentioned, the disease starts with this relapsing early phase, for which there's pretty good medication on the market right now. However, the progression in the late state of the disease is still really unmanageable, and we really lack effective medication tackling that progression. And that also partially is driven by a lack of understanding of the exact molecular mechanisms and pathologies that drive this progression. Now, the white matter lesion pathology in MS is quite diverse. As Bart already mentioned, there's these different lesion types, active lesions and mixed active and inactive lesions with these cores and rims. However, I want to put one more layer of separation on top of that, is that we also have now classified lesions according to the morphology of the microglia that you find in these active lesions or mixed active inactive lesions. And Bart already mentioned these foamy microglia, that they arise in MS. You can really see them by the rounded morphology and the lipid-laden appearance. You see these vesicles arising in the cytoplasm of these microglial cells. So that's also why they're called lipid-laden or myelin-laden. And we've known that they arise for a long time, but what their exact role is quite unclear. Are they beneficial or detrimental? Are they a determinant for the outcome of this lesion? That still remains a question. What we've seen in the clinical cohort of the Netherlands Brain Bank is that MS donors who at the time of their death had a lot of lesions with foamy microglia in their white matter actually tended to have a very progressive disease. So we see that donors with a lot of foamy lesions have a shorter time to reach clinical milestones like the EDSS 6 and 8 scores, but they also have a shorter disease duration overall, and they die at a younger age, which indicated to us that they had a very fast disease progression. So we hypothesized that these foamy microglia are probably a bad sign. They are detrimental to the lesion progression. And our main research questions were to identify the molecular mechanisms of foamy microglia in MS lesions, and if we could find druggable targets that target lipid metabolism to potentially resolve these lesions. So in order to do this, we obtained tissue from the Netherlands Brain Bank, and we isolated all different lesion types available, but now in addition we have also analyzed lesions with more ramified microglia or foamy microglia. In total, we analyzed 110 brain samples with extensive histological analysis. We characterized these lesions for all the different cell types. And then I want to make a nuance here that when we classify a lesion in either non-foamy or foamy, this is not 100%. It's not like the 100% of the microglia are foamy and then the non-foamy are 0%. But when we quantify these number of foamy microglia, we see that in lesions we classify as foamy, there's on average more than 50% of the microglia are foamy, whereas in the lesions we classify as ramified or non-foamy, this percentage is really down to almost 0. So it's more the presence of foamy microglia within an MS lesion. Now, this wouldn't be a cross-omics festival if we didn't then start to do many omics technologies, including lipidomics, chemical proteomics, global proteomics, and gene expression analysis. But because of the central role of lipids in these foamy microglia, I want to take you through the lipidomics data first. So in collaboration with the labs of Thomas Hankemeyer and Amy Harms, with postdoc Qin Yu, who measured all these lipidomics platforms, we have measured the 712 lipid species across 31 different lipid classes. And there's a lot of data to go through here, but I want to focus specifically on the foamy microglia, because we really see that these really separate from the other lesion types. And what is driving this separation is mainly driven by cholesterol esters that we see accumulating in these foamy microglia. So these the cholesterol esters are really a marker of lesions that contain foamy microglia, but we also see increases in triglycerides, and interestingly we see increases in bismonoacylglycerolphosphates, which are lysosomal lipids, which indicate that these microglia have a higher lysosomal load, which could indicate more phagocytic functions. To confirm this cholesterol ester load, we did oil red O staining, which really stains the neutral lipids in these types of lesions. You can really see that the red dots appear only in the rims of lesions with foamy microglia, but are basically absent in lesions with more ramified microglia. And then you can also see that these really resemble these vacuoles of probably cholesterol ester-filled lipid droplets. Another class of lipids that was really closely associated with foamy microglia were oxidized free fatty acids. We call them oxylipins for short. And those are the products of oxidation of free fatty acids, and they're generally regarded as pro-inflammatory mediators. So that indicates also that there's a different inflammatory signature in lesions with either foamy or non-foamy microglia. To complement these lipidomics analysis, we also did transcriptomics, where we quantified almost 17,000 genes, and proteomics in collaboration with the lab of Albert Heck. Tatjana did these measurements, and in both analyses we could see that the foamy lesions with foamy microglia also have a distinct profile. We see them separating in analysis like principal component analysis. And we see many genes that also match our lipidomics data. So we see genes upregulated in the formation of oxylipins, lipid droplets, lipid peroxidations, and cholesterol homeostasis versus recycling. We see a shift from recycling to from synthesis to recycling. So that really matches closely our lipidomics analysis. However, as Bart already mentioned, the bulk analysis has its limitations. That we lose the spatial and cellular resolutions of our analysis. People generally, you could compare this to the smoothie versus the fruit salad, where we now only see the complete mixture, and then the question arises, are the foamy microglia really different, or are we just looking at lesions with much more of this, or more microglia in general? So the advantage of doing bulk RNA sequencing is the cost effectiveness, so we could do a lot more samples, and based on previous single nuke RNA sequencing data sets, we could then try to deconvolute our bulk data. So we have obtained a single nuke RNA sequencing data set from this recent paper that also did transcriptomics on MS lesions, and then relate this to our data set. So basically we could use deconvolution to estimate the cell type abundance in our samples, and we could, for example, see that oligodendrocytes, the cell type making myelin, really drastically reduced in all lesion types. And we see, as expected, also an increase in the number of T and B cells, for example. But now we're focusing mainly on the microglia, and although microglia are increasing from roughly 10% to 20 to 30% in lesions, There's no significant difference between foamy and non-foamy microglia containing lesions. And if we correct our analysis for the number of microglia, we actually see that many of the genes that are differentially regulated between foamy and non-foamy containing lesions, most of the significantly altered genes remain the same even if we correct for the number of microglia, which suggests that really is expression by the microglia themselves, not just the cell type composition itself. To go a little bit deeper into the states that were described in the same paper, so this paper also described many microglial states, some of which are overlapping with the states Bart already showed you. And we really see that one specific state, in this case the micro D cluster, is really strongly associated with lesions with the foamy microglia. And this is also confirmed significantly increased in foamy versus non-foamy. And interestingly, the more homeostatic cluster, micro A, is really reduced in lesions with foamy microglia. So they're less homeostatic and more shifted towards this micro D state. If we look at the marker genes that really characterize this state, the top gene is GPNMB. And interestingly, GPNMB was also like one of the top hits in our differential expression analysis between foamy and non-foamy containing lesions. For proteomics, even very clearly the marker gene. So staining that also confirmed that these microglia that are, I hope you can appreciate the difference in morphology here between the foamy and the more ramified microglia, and that the GPNMB is much higher expressed in those lesions containing foamies. And also they really localize to the lesion rims. So Bart already showed you that these lesions have an inactive core, and then from the rims they expand, and these GPNMB positive microglia really accumulate at the rims of those lesions, suggesting that they're involved in the expansion of these lesions. Now, with all these different omics data in hand, we could also form like an integrated molecular profile using multi-omics factor analysis, which basically looks at the covariance in the different data modalities and derives shared factors. And one of factor 1 and 2 were mainly associated to general lesions, so whether there was a lesion or not. But for factor 3 really was selected for the foamy phenotype, and also significantly increased in lesions with foamy, and also, interestingly, very closely correlated with the signature of this micro D microglial state. If we now correlate these factor values back to the clinical progression data of the patients where these samples came from, we do see correlations between the score on factor 3 and the same progression markers that we use to assess their clinical trajectory. And we see that samples that have a high factor 3 score almost exclusively come from patients with a very fast disease progression. So the first question I asked was what's the molecular profile of these foamy microglia? I think we can answer that. They're really characterized by this cholesterol accumulation in lipid droplets. They have increased lysosomal content and stress. They produce these inflammatory oxylipins, and we can couple them to a specific transcriptional state using single-cell reference data sets, which here are characterized by the GPNMB protein. And we think they are likely involved in the expansions of these lesions. Next question is then, can we identify druggable targets? And maybe to refine this question, can we specifically identify druggable targets in foamy microglia? To do this, we turn to a specific family of enzymes called the serine hydrolases. So these enzymes are really closely involved in lipid metabolism. They do this with this active serine residue in their pocket, and they cleave ester bonds in lipids. And we have a trick to basically capture these enzymes when they're in an active state. They react with this fluorophosphonate moiety, and we can basically trap them. And what we can do then is using probes like this, we can enrich the active enzymes from a mixture, analyze them by mass spectrometry, and get a view of the metabolic enzymes in these MS lesions. So we did that, identified 97 enzyme activities, and then compared foamy to non-foamy containing lesions, and found that many metabolic enzyme activities were higher in these lesions containing foamy microglia. One of them, interestingly, the lysosomal enzymes were very prominent across this panel of enzymes, which matches the lysosomal phenotype we described previously. But interestingly, the monoacylglycerolphosphates was one of the enzymes popping up here, and that caught our attention because a MAC lipase is involved in degrading the endocannabinoid 2-AG to a free arachidonic acid, which is then the precursor for oxidation for pro-inflammatory production of prostaglandin E2. And MAC lipase is basically the node that controls both 2-AG and oxylipin levels in the brain. Interestingly, we also see that lesions with foamy microglia have a reduced 2-AG level, and as I previously showed you, the oxylipin levels are increased in those lesions with foamy microglia. So we really think that the MAC lipase is an important node in this metabolic switch from 2-AG to pro-inflammatory oxylipin production. We could also confirm that with RNA scope analysis, we see that the expression of MAC lipase is basically absent in normal control white matter, but is then upregulated strongly in lesions, also specifically in microglia, and we see that they are the highest in mixed lesions with, in the rims of mixed lesions with foamy microglia. We also developed this probe that directly visualizes active MAC lipase in tissues, and we can use that to directly show where MAC lipase is active in a postmortem tissue. So we see that in this rim of a mixed lesion, we also see a lot of MAC lipase activity, specifically localizing also to foamy microglia, but also to some ester sites, reactive ester sites that are found in and around these lesions. With that, I want to draw some conclusion. I think the cross-omics profiling here of MS lesions delivered a very comprehensive map of specifically the foamy microglia, because that really was a big driver of variability in our data set. Foamy lesions associated with neurodegeneration and also with increased lipid oxidation, cholesterol ester accumulation, and lysosomal stress. And lastly, I think our data will support the development of MAC lipase inhibitors for the treatment of progressive MS. With that, I want to thank all the people involved at Leiden University, at Utrecht University, at the Netherlands Institute for Neuroscience, at Roche, and most also the Netherlands Brain Bank. I want to thank the donors and their relatives for their courageous contribution to science. And I want to thank you for your attention. And if there's any questions, I'll be happy to answer them.